Hello everybody. I'm very grateful for this opportunity to speak with you about some of our research and to show you how some of our findings may have applications in your own work. The title of my presentation is Using Insects to Assess the In Vivo Toxicity and Activity of Antimicrobial Agents. And in this presentation, I hope to be able to show you how we can use insects instead of mammals for routine in vivo screening of novel compounds, and that insects offer an alternative way to assess toxicity and in vivo activity. So the first question we have to answer is, why do we need in vivo screening systems? Well, before we can develop any drugs for use in humans, we need to assess their efficacy in an in vivo system, usually in mammals. We may need to examine how the drug interacts with the host's immune system or the host's biology, and if there are adverse effects, the drug or compound can't be developed further. We may wish to establish how the drug is metabolized. Maybe it's metabolized to toxic side products, which would obviously have adverse effects. Using in vivo systems, we may be able to identify novel targets for therapy to help us fine-tune new therapies. And obviously, before we can embark on clinical trials, routine in vivo screening using a range of mammals is required. So in vivo screening systems are essential to ensure safety, to measure toxicity, and to measure efficacy of compounds. A range of mammals, a range of other vertebrates are used. So a range of mammals are used as in vivo screening systems. The most widely used would be mice, guinea pigs and rabbits, but rats and dogs are also used in some applications. Sometimes primates are used, such as monkeys and apes, and human volunteers. Sometimes birds can be used for particular applications, such as pigeons and chickens, and occasionally reptiles are used, depending on the application. So within the EU each year, approximately 8 million vertebrates are used in vivo screening systems. And in Ireland, this figure is around about 250,000, mostly small mammals, mice, rabbits and rats. So a lot of mammals, a lot of vertebrates are used routinely in this type of testing each year. Why is this? Well, we need to establish tolerability in a mammal before testing in humans. We need to determine how effective a drug is and establish a dosing regime. There are distinct disadvantages, however. For example, it's extremely expensive to do this work. Even routine testing with mice or with rats costs thousands of euro. Typically, the work is very slow. It may extend over a number of weeks to get results, and it's also ethically questionable. Are we right to use so many mammals, in some cases inflicting suffering on these mammals to get results? However, the testing using vertebrates is mandatory prior to drug development and release for use in humans. But everybody recognises we should try and minimise this or refine it as much as possible. As a result of these concerns, there has been a move to develop alternative systems, and this has been encapsulated by the 3R policy. So this policy aims to replace, reduce, or refine the use of vertebrates in testing. And it's now a recognised policy, and it has been adopted by EU, by governments and by funding agencies. And in many cases, funding agencies will ask applicants to show how their work complies with the 3R policy. This is a good move, and it is welcomed by all in the scientific community, but an alternative system, whatever it is, must be valid and effective. And it must give us comparable results to those that we might be able to obtain using vertebrates. So, over the years, a number of alternative systems have been developed, and each has its own advantage. But I'm going to focus on one particular system and show how it can provide information to replace the need to use mammals. So, insects are an extremely successful group of animals. They exist in vast numbers and occupy almost every ecosystem on the planet. The one place they have not conquered is the sea. They can't survive in the sea, but every other ecosystem, every other niche, insects can colonize. Why is this? Well, they can reproduce at a very fast rate, producing hundreds or thousands of offspring quite rapidly, but they also have a very successful immune system. And this immune system allows them to identify a pathogen very quickly and either kill it directly or immobilize it so it can't develop. This immune system is very similar to our innate immune system. So, using this information, we can then begin to think about using insects to model how pathogens might interact with our innate immune response. So just to give you an example of how similar the insect immune response and the human innate immune response are, we can take cells from both systems. And in this case, we have an electron micrograph of a human neutrophil, and this is an important cell in the innate immune response. It protects us from pathogens. So the neutrophil takes in bacteria or other pathogens and digests them and kills them. So when we look down the electron microscope, we can see a number of features of a neutrophil indicated. We can see internally dark coloured structures. These are called granules, and they contain enzymes that help to chop up pathogens when it's been taken in by the neutrophil. And then the arrow indicating structure 3, this is a pseudopodia, which helps the neutrophil to entrap a pathogen to be killed. The image on the right is an immune cell from an insect, and this is known as a hemocyte. And again, in the hemocyte, we see granules which contain enzymes which do the killing, exactly as we see in neutrophils. And we also see evidence of pseudopodia, which are indicated by the arrow marked number 3 in this picture also. So both of these cells structurally look very similar, and they both function in the same way. They take in pathogens and then they digest the pathogen and kill it. If we didn't have neutrophils, or insects didn't have hemocytes, we would become overrun by pathogens and they would kill us. So these are essential cells. But as we can see, the human neutrophil and the insect hemocyte are extremely similar, structurally and functionally. So another level of similarity can be shown in the way in which insects and mammals detect pathogens that have entered the body. So once a pathogen enters the body, there are a number of receptors which can pick up parts of the pathogen, and these then can trigger a response to fight off the pathogen. So how do insects respond? Well, this is shown on the left-hand side by the toll receptor in invertebrates. So toll is a protein that sits in the membrane, and once it becomes activated, it initiates a cascade of reactions that causes a transcription factor known as diff to cross into the nucleus. Once it's in the nucleus, it turns on certain genes, and these will lead to the production of antimicrobial peptides or antifungal peptides to kill the pathogen that has entered. The corresponding system in vertebrates or in humans is extremely similar. So in this case, the protein that sits on the membrane is known as TLR, and once it is activated by the presence of material from a pathogen, it too initiates a cascade of reactions which causes a transcription factor, in this case known as NF-kappa beta, to pass from the cell cytoplasm into the nucleus to turn on the expression of certain genes to deal with the pathogen. So, at a number of levels, the invertebrate system and the vertebrate system for identifying pathogens is extremely similar. They've both originated from the same ancestor. So, when we look at the functioning of the insect immune response and the innate immune response of mammals, we can see they're extremely similar. Similar types of cells, similar types of receptors, similar types of antimicrobial peptides produced. So very similar at many different levels. And as a result of these similarities, a wide range of insects are now used as in vivo screening systems, and these are often referred to as mini models. So one of the most widely used, and the subject of what I'm going to talk about today, is the insect Galleria mellonella. This is the wax moth. In the wild, it's a pathogen of beehives, but it can be cultured in the laboratory and is very easy to use and easy to get results from. And I will demonstrate some examples of this. One of the most famous insects in biology is Drosophila melanogaster, and this has been one of the most important animals used in biology and developmental biology over the last 50 years, particularly for studying genetics. So it's useful because a range of mutants exist of Drosophila, but the problem as a screening system is it's difficult to inoculate because they're so small, but they do have some applications. Manduca sexta, this is the tobacco moth, and these are quite large insects, and they can be cultured in the laboratory, and we can use them for isolating immune cells, but they have an application as a source of cells mostly. And then Bombyx mori is the silkworm, and it is now used for screening antimicrobial agents, for screening the toxicity of antimicrobials, or for screening their efficacy. So, as a result of the conserved similarities between the insect immune response and the innate immune response of mammals, a number of insects are now available as in vivo screening systems. So as we'll see, these can be used in place of mammals, and they give comparable results to the results that can be obtained using mammals. The larvae or caterpillars of Galleria mellonella are now widely used in research laboratories, both academic laboratories and industrial laboratories. And they're easy to work with. So they can be inoculated in two ways. One is by intrahemocoel injection, which involves injecting in a compound or a pathogen directly into their body cavity. So this would be similar to us getting an injection into our blood system. The other possibility is to feed them. So we can hold the larvae in such a way that its mouth opens and we can give them a small dose of a compound or a product, and then we can look at how they will metabolize this. So this image shows on the left hand side healthy Galleria larvae. So they are sort of cream in colour, they're quite active, they move around, but if they're injected with a pathogen called Aspergillus fumigatus, which is a fungal pathogen, over 24 hours they darken in colour and die. So we now know that this pathogen, Aspergillus fumigatus, is highly virulent and can kill the larvae. It's quite clear the ones on the right hand side are dead. So this gives us the possibility to uncover how this pathogen interacts with their immune response and how it succeeds in overcoming their immune response in a similar way that it does in vertebrates. So we can now see that the insect immune response is very similar to the innate immune response of mammals, and in mammals the innate immune response is the first line of defense against pathogens. So what I'd like to do now is to show you some examples of where we can use insects instead of mammals to get comparable results. So one pathogen that we deal with is the yeast Candida albicans, and Candida albicans exists on our bodies, and for most of the time it's not a problem, it's non-pathogenic. However, if our immune system becomes weakened, it becomes pathogenic, and in extreme cases it can kill. So it is a dimorphic yeast, that is, it exists in two distinct morphologies. On the left-hand side, we can see the budding cells, and it grows like this if we grow it in the laboratory. But under certain conditions, it begins to produce hyphae, which are thread-like structures and can invade tissue and colonize the body. This form, the hyphal form, tends to be the more pathogenic or more capable of causing disease. So if we have a mutant that can't form this type of growth but stays in the yeast form of growth, it will be less virulent. So this slide shows the results of experiments looking at the virulence of Candida albicans mutants when measured in mice and when measured in Galleria larvae. So if we look at the image in the upper left-hand side, this is an experiment which measured the virulence of a mutant strain and a parental strain in mice. The mutant strain in this case can't form hyphae. It can't form the hyphal growth form. And as you can see, none of the mice infected with this strain died over 42 days. Mice that were infected with the parental strain, which could form hyphae, were all dead at 15 days. If we do this same experiment in larvae, we can see that the wild type strain, which is CA-SC5314, kills all the larvae in 24 hours. The mutant strain, which is CR216, doesn't kill any in 48 hours. Another experiment uses a strain which can form hyphae but at a very low rate. So again, on the left hand side, mice are infected with the mutant, and this results in the death of about 50% in 21 days. Mice infected with the parental strain, which rapidly forms the hyphal form of growth, results in death at 6 days. So we can see the mutant isn't avirulent, but it's not as virulent as the parental strain. If we do that experiment in Galleria, we find the parental strain kills all the larvae by 24 hours, but the mutant only kills about 50% at 24 hours and kills about 90% at 48 hours. So again, demonstrating that the mutant is not avirulent, but it retains some virulence. So this series of experiments demonstrated a strong similarity between the response of mice to Candida albicans mutants and the response of larvae to the same mutants. And in the full study, we could demonstrate a correlation in 90% of cases. The other thing to note is that the work with mice lasted between 21 and 42 days to get results. In contrast, when we were doing the experiment in Galleria larvae, we were getting results within 2 days. Another point to emphasize is the cost of doing this experiment in Galleria larvae would have been less than €1 or €2. The cost of doing the experiment in mice would have been of the order of tens of thousands of euro. Another way we can use the larvae is to use them to study how disease develops in the insect. So see how the immune response interacts with the pathogen and then see how one predominates. So in this case, larvae have been infected with Aspergillus fumigatus, which is a fungal pathogen, and we can observe that larvae that are infected with 1 by 10 to the 6 spores of this pathogen results in about 90% death at 72 hours. If we inoculate them with 1 by 10 to the 7 spores, we have 100% kill at 48 hours. So this pathogen is extremely virulent and kills larvae. It's also a pathogen in humans. But we can go beyond just looking to see if it does or doesn't kill the insect. We can study what's happening inside the insect. And in this case, we have lateral sections through larvae using a cryovis, and we can visualize the dissemination of the pathogen through the insect and the development of disease symptoms over time. So, for example, the larvae at the top is at 0 hours. It has just been inoculated, and the point of inoculation is shown by the arrow with the white border around it. Then by 6 hours, when we look at the larvae, the black arrows indicate areas where the pathogen has been spread to within the body. So it very rapidly disseminates around the body, and then by 24 hours, we can see the development of structures known as granulomas. These are large structures that develop where the immune system tries to fight off the pathogen. And we can also see that the skin or the cuticle of the larvae begins to darken in colour, which is a process known as melanisation. So importantly, in this image, we see the development of structures called granulomas. we can remove granulomas from the infected larvae and stain them with calcifluor, which identifies fungal material, or FM464, which identifies membranes of immune cells from larvae. So we can identify that the granuloma is made up of a composition of fungal cells and hyphae, and also immune cells of the larvae. So this structure, this type of structure, is also seen in human cells and human tissues that are infected by Aspergillus fumigatus. Again, giving us another insight into the similarities between the response of the insect and the response of humans. We can extend this study further by looking at how the insect responds to the pathogen and then establishing similarities between the insect response and the mammalian response. So in this case, we can do label-free proteomics. So we can take samples of blood or hemolymph from the infected larvae at the different time points, and then we can analyse these by mass spectrometry and identify the peptides present in the hemolymph at different time points. So for example, we can establish the type of larval proteins that are increased in abundance, and we can also establish the type of fungal proteins that are altered in abundance. And we can see how the two systems, the pathogen and the host, interact together during the development of disease. So another widely used application of Galleria larvae is as an in vivo screening system for measuring the efficacy of antimicrobial agents. These may be conventional antimicrobial agents or novel agents that are recently being synthesized and are being characterized. So in this scenario, the larvae can be infected with a variety of pathogens, gram-positive bacteria or gram-negative bacteria, or a range of fungi. These can then be followed up with the antimicrobial agent of interest, and we can measure the response of the larvae. So we can use a variety of endpoints. We can measure survival. We can measure the extent of melanisation. How dark do the larvae go? We can study changes in the immune cells in the larvae, which tells us how good they are at fighting off the pathogen. We can look at the proliferation of the pathogen inside the larvae and see if the newly introduced antimicrobial drug is inhibiting its development. And then we can apply a variety of proteomic techniques or histopathological techniques to study the development of the pathogen and to characterize how the host is responding to it. So as a result of this development, larvae are now widely used in screening novel drug candidates, and it can inform subsequent synthesis to optimize toxicity or to optimize efficacy in vivo. So we'll see some examples of this shortly. This figure describes the result of experiments to assess the efficacy of a number of silver-based complexes in inhibiting the development of Candida albicans when it is used to infect larvae. If we look at the control, these larvae were infected with Candida albicans, and we can see that by 72 hours, around about 90% have been killed. However, if we infect the larvae and then follow up with a dose of silver malfen, we can see protection and increased survival at 72 hours. Equally, if another batch of larvae are infected with Candida albicans and then followed up with a dose of silver nitrate, we get approximately 40% survival at 72 hours. And then if we use another group and follow up with phenanthrene, we also get protection. So in this case, we can demonstrate that the silver complexes can give protection against Candida albicans development. So we know the silver complexes have antifungal activity, and we can demonstrate this in vitro assays in the lab. We can also demonstrate they operate in vivo in the Galleria larvae. But they also do something else, and this is demonstrated in this graph. So, in this case, we've measured the density of hemocytes in the larvae. And remember, the hemocytes are the immune cells in the larval response. And we can see in the control there's no change in the hemocyte density. However, in those that receive the silver nitrate, the silver malfen or the phenanthrene, there is a significant increase in the hemocyte density. So as well as exhibiting antifungal activity, the complexes also induce an increase in the density of circulating immune cells. And these also contribute to the antifungal activity. These complexes also trigger an increase in the expression of genes coding for galeromycin, a potent antifungal peptide produced by larvae. So in this case, we can see that the complexes have antifungal activity, but they also trigger a response in the larvae. They trigger an increase in the density of circulating immune cells, and they trigger an increase in the expression of a particular antimicrobial peptide. And both of these factors influence the development of Candida albicans in vivo. We have also performed work with another silver-based compound with the group of Professor Matthias Tack in UCD. And this compound is codenamed SBC3. And it is a potent antibacterial agent, and we can assess this in the lab. And we can see that at a dose of 100 microgram per mil, we inhibit the growth of Staphylococcus aureus by about 70%. So, what's the in vivo situation? So in this case, larvae were infected with Staphylococcus aureus, and we can see in the control strains approximately 80 to 90% were killed by 72 hours. However, if we infect larvae with Staph aureus and follow up with SBC3, we find a reduction in the kill. So, for example, those larvae that received a dose of 10 micrograms per mil after infection with Staph aureus, we see approximately 50% survival at 72 hours. So, this works in vivo. A similar approach was followed using mice that were infected with MRSA. And MRSA stands for methicillin-resistant Staphylococcus aureus. In this case, mice were infected systemically with MRSA, and then after 24 hours they were euthanized and their thigh muscle was removed, and the number of bacteria in the muscle was quantified. So in the control strains, those that received MRSA and no treatment, we find a MRSA density of 1 by 10 to the 10 cells in the thigh muscle. Those mice that received the dose of SBC3 subsequent to infection show a reduced density of bacteria in the thigh muscle. So, for example, those that received a dose corresponding to 40 mg per kilogram showed a bacterial load in the thigh muscle of 1 by 10 to the 9, so tenfold less. So again, we see in this case, the SBC3 can be demonstrated to work in vivo using Galleria larvae, and in this study, it was also demonstrated to reduce the burden in MRSA infected mice. We have recently begun to study gallium complexes with Dr. Darren Griffith of RCSI. And in this case, we are interested in the ability of gallium maltolate to inhibit the growth of Pseudomonas aeruginosa. So on the left-hand side, we can see the effect of gallium complex on the growth of Pseudomonas aeruginosa, and we can see at a dose of 250 micrograms per mil, we've reduced growth by approximately 75%. We can also infect larvae with Pseudomonas aeruginosa, and this is a very potent bacteria that is very virulent in insects. So if we infect them with a dose of 3 by 10 to the 2 cells or 3 by 10 to the 3 cells, we have 0% survival 24 hours later. However, if we infect them with that dose of bacteria and then follow up with a dose of gallium maltolate, we find increased survival irrespective of the infecting dose. So again, we can demonstrate that the complex works in vitro in the laboratory experiment and in vivo in the larval system. So another application has been the use of larvae for examining the metabolism of compounds, and this can give us an indication of their toxicity or metabolism in mammals. An example of this application has been to look at the relative toxicity of a range of food preservatives. And as we know, food preservatives are widely used, but they are toxic above certain concentrations. So their use needs to be within certain limits. So in one series of experiments, we measured the toxicity of food preservatives in larvae, and we compared this with the toxicity of the same compounds in mammals. And the results of this experiment shows a strong correlation between the toxicity of the food preservatives in Galleria larvae and the toxicity of the agents in rats. On the y-axis, we have the LD50 of the agents in larvae, and on the x-axis, we have the corresponding LD50 values in rats. And as we can see, we have been able to establish a correlation. So this means that we can assess the toxicity of food preservatives in larvae and get a reasonable estimation of their relative toxicity in rats. So this type of work can be extended to look at the metabolism of compounds in larvae. So in this case, larvae have been fed caffeine, which as we know is a stimulant, and the metabolism of it has been followed by HPLC. And in the upper graph, we can see the breakdown of caffeine in the larvae over time, and we can see corresponding increase in the breakdown products, the theobromine and the theophylline in the lower graph. And as well as measuring the metabolism of the caffeine, we can also measure its effects. And one of the effects of caffeine administration on larvae is that it is a relaxant. They become incapable of moving. And this is demonstrated in this graph. So larvae in this case have been given caffeine, and then if they're turned on their back, we measure the length of time it takes them to straighten or to right themselves. So this is measured in seconds on the y-axis. So we can see initially they can right themselves quite quickly. However, by 4 and 24 hours, they are very slow to right themselves. And then eventually they regain the ability to move at a normal rate by 72 hours. So this inhibition parallels the breakdown of caffeine to its metabolites. We can also use the Galleria larvae to measure the response to novel compounds. So in this case, we were looking at the response of larvae to a number of copper complexes, and structures of which are shown here. This work was done in collaboration with Professor Orla Howe and Dr. Andrew Kellett. And we were able to establish the in vivo response using Galleria of these agents which are capable of intercalating with DNA and disrupting DNA replication. And one approach that was taken was a proteomic approach to examine how the larvae were responding to the different copper-based complexes. So hemolymph was taken from the larvae. This was processed for mass spectrometry, and then the data was analysed by Blast2Go and by KEGG analysis. And the results then give us an indication of the response of the larvae, but also point to the likely response of mammals to administration of these complexes. And one set of results that we got can be displayed as volcano plots, showing the proteins increased in abundance or decreased in abundance following administration with the copper-based complex. And we can see, for example, that the greatest change in protein abundance occurs when the larvae were given the copper DPPZ-Fen complex, and a range of proteins have been statistically increased in abundance and a number are decreased. So again, giving us an idea of the metabolism and the response of the larvae. And using this analysis, we can identify the principal proteins altered in abundance following administration of the copper complexes to the larvae. So we can see, for example, a range of stress proteins are increased in abundance, proteins in glycolysis can be increased, proteins involved in purine metabolism can be increased in abundance. And the relative fold increase in these following administration of each complex is shown here. So this result then allows us to identify which is the most active complex, how does the larvae respond to it, and it gives us in vivo insight into the likely response of a mammal to these complexes before we move to mammalian testing. So I have given you some examples of how Galleria larvae can be used as an in vivo screening system. They can be used to measure the virulence of a pathogen or a mutant, to assess the relative toxicity of novel complexes, to measure the in vivo activity of an antimicrobial agent, or to study how a complex is metabolized. And they can give results that are comparable, maybe not identical, to those that may be obtained using mammals. So clearly, they have potential applications. So the Gallerial system has many advantages. As I've mentioned earlier, they're easy to purchase, to house and to use. They're relatively cheap. So a box of approximately 200 costs around about €8. You can get results within 48 hours. Comparable mammalian testing can take up to 6 weeks. You can screen a lot of compounds very quickly, depending on what you're looking for. And there are a range of endpoints that you can use. So you might want to measure survival, you might want to measure changes in the proteome, you might want to measure some other factor, but all of these are possible. And they've parallels with endpoints in mammalian testing. The larval system is ethically acceptable. So many funding agencies will require evidence that the application is using the minimum number of mammals and using an alternative comparable system, such as Galleria larvae. As I've said, the results are comparable to those you can get from murine testing, and many people are now using Galleria larvae as a pre-screen system to identify the most potent complexes, or those complexes with the lowest toxicity, before embarking on the more expensive and ethically challenging mammalian testing. And another way larvae have been used is to inform synthesis. So to screen some compounds in larvae, to look at the response, and then to move to change synthesis to alter some factor or element of the new complex to reduce toxicity, for example, and then retry that. So any system obviously has disadvantages, and this is true in the case of the Gallerial system as any other. So one disadvantage is that confirmatory testing in the murine system is invariably needed if the product is to be further developed. But you might need to use as many mice as you would otherwise. The larvae also lack many vertebrate-specific systems. So, for example, they don't have lungs, they have a different type of digestive system. So that is a definite disadvantage, but they have systems that we can still study. The larvae are not fully sequenced, so as a result, there are no mutants available, unlike in the case of Drosophila, for which there are a range of particular mutants. So I suppose, what's the take-home message? If you're interested in vivo screening or in vivo testing, the standard up until recently has been to use mammals, but everybody wants to use the number of mammals, both from a cost point of view and an ethical point of view. There are alternative systems now available, and one of these is the use of insects, particularly Galleria larvae. They have a range of applications. They may be useful to your work, particularly if in the medium to long term you see your work progressing to use mammals. They can fine-tune the product, fine-tune the synthesis, so to maximize efficacy when you do move to mammalian testing. So before I conclude, I would like to thank you for your attention and also to thank the various people who've worked on this work over the years, the postgraduate students who have done the work, and also my collaborators who have assisted at various stages in developing this system. Our proteomic facilities were funded by Science Foundation Ireland, and I'm grateful to them and also to the SSPC for continuing our work on developing this system. If you need more information or if we can help in any way, please feel free to contact me and I'd be delighted to assist you. Thank you very much.